Good afternoon, everyone. Am I audible? Okay. So, again, let's start today's session. This will be the second tutorial session wherein I will be uh, summarizing the week 2 lecture videos, whatever was taught in the lecture videos, and then we will try to solve some assignment questions. Okay. And so I will start sharing my screen and just let me know, because once I start presenting I won't be able to know who all are joining. If someone is waiting to get like joining to join the call, then just let me know, just unmute your mic and let me know, then I will come back and admit that person. Okay, so let's start present. Okay, is my screen visible to everyone, like the presentation, the screen? Okay. Okay. So this week's first few lecture videos were regarding what all reaction go on inside a cell to get, to procure energy or to build up biomolecules. So the word that is used is metabolism, which refers to all sorts of reactions that go on inside a cell. And metabolism can be divided into two, basically like broadly divided into two sections called anabolism, which refers to the biosynthetic pathways which actually build up complex molecules using energy, and catabolism refers to those pathways or reactions in the cell or inside our bodies, not only our bodies, in living organisms, which use energy, which can break down complex molecules and give us energy. Okay, so maybe just we will go through the role of ATP, which acts as an intermediate between catabolic and anabolic reactions. Okay, so catabolic reactions here transfer energy from complex molecules to ATP. Okay, and then it forms, when heat is released, energy is released, then it forms simple molecules like glucose, amino acids, which actually are joined together to give us proteins or peptides, then glycerol, fatty acids, and then these simple molecules can again be used to for anabolic reactions as reactants for anabolic reactions, which transfers energy from ATP to complex molecules. Again, heat can be released, thereby forming complex molecules like starch, proteins, and lipids. Okay. Now, there we looked at glycolysis, the glycolytic pathway, which basically uses glucose to generate ATP. Now, glycolytic pathway can be again broadly divided into two phases: preparatory phase and payoff phase. Preparatory phase is this first half preparatory phase wherein energy is actually required, whereas payoff phase is where energy is released in the form of ATP, and also there are certain NADH molecules are also released, which are later processed in the subsequent pathways or downstream metabolic pathways, which can again give us ATP, which can produce ATP molecules. So maybe I will zoom this a little bit. Huh. So the preparatory phase, what is there? First, you have glucose, which uses ATP, which uses one phosphate from, this is a triphosphate, one phosphate from ATP, releases diphosphate and uses that phosphate to phosphorylate this glucose molecule, forming glucose 6 phosphate. The enzyme responsible here is hexokinase. Okay, then from glucose 6 phosphate to fructose 6 phosphate. So what is basically happening here? Glucose and fructose are isomers. Okay, so the enzyme that will be used is isomerase. Okay, so and hexose refers to 6 ring uh, substrate in glucose and fructose, 6-membered carbon ring. Okay, so isomerase is used for changing as in for converting glucose 6-phosphate to fructose 6-phosphate. Then again one ATP is used to phosphorylate one more residue in fructose and forming fructose 1,6 bisphosphate using phosphofructokinase. Kinase is any type of enzyme which phosphorylates any type of residue or substrate is called kinase enzyme. And then what all substrate is being phosphorylated, that depending on that the name changes, the nomenclature can change. Okay, next, this fructose 1,6 bisphosphate used ATP. One another molecule of ATP was used, and this was formed glyceraldehyde 3-phosphate and DHAP or dihydroxyacetone phosphate. So till here we had 6-membered substrate. Now it changes to, see, 3-membered substrate each. Glyceraldehyde 3-phosphate 3-membered, I mean 3-carbon substrate, and DHAP 3-carbon substrate. Now next we have payoff phase, wherein you have we actually get ATP molecules from these reactions. Now so all these reactions are showing, so this 2 refers to, because one 3-carbon substrate, another 3-carbon substrate, when both of them are subsequently metabolized, then you ultimately have a total of 2 molecules which are getting used up or released, either ways. So what is happening in payoff phase is oxidative conversion of glyceraldehyde 3-phosphate to pyruvate, which is the end product of a glycolytic pathway, and the coupled formation of ATP and NADH. So glyceraldehyde 3-phosphate forms 1,3-bisphosphoglycerate. NADH is released. The enzyme here is glyceraldehyde 3-phosphate dehydrogenase. Okay. Next, again, 3-phosphoglycerate is formed. See, 1,3-bisphosphoglycerate, 2 phosphorus were present. Phosphorus atoms were present. Phosphate groups were present, and one group was released, which was used to form ATP, thereby forming 3-phosphoglycerate. The enzyme used was phosphoglycerate kinase, and then again 3-phosphoglycerate to 2-phosphoglycerate. This enzyme plays the role in catalyzing this reaction, phosphoglycerate mutase. Then from 2-phosphoglycerate, water molecule is released to form phosphoenol pyruvate or PEP using enolase enzyme, which is this reaction is catalyzed by enolase enzyme. Then PEP is finally converted to pyruvate using pyruvate kinase, and one ATP molecule is released for one molecule of glyceraldehyde 3-phosphate. Okay. So here oxidation and phosphorylation is taking place, and this is the first ATP forming reaction. 1,3-bisphosphoglycerate to 3-phosphoglycerate, one ATP molecule is released for 3-carbon, one 3-carbon substrate. And next second ATP forming reaction is from PEP to pyruvate. Okay. Oh. Hmm. Next, so the balance sheet for glycolysis is input 1 glucose molecule, 4 ADP plus 4 inorganic phosphate molecules, 2 ATP in the preparatory phase, 2 ATP molecules and 2 NAD plus molecules. And output, what we are getting, 2 pyruvate molecules from glyceraldehyde 3-phosphate or DHAP, 2 pyruvate molecules. Net production of ATP is 2 and 2 NADH molecules. Now, the lecture videos had covered the portion containing how glycolytic pathway is regulated. So maybe we can just look at it one by one. So what was happening is it is regulated by insulin. What happens is insulin is uh, mainly required to regulate the levels of glucose in the body or glucose in the blood. So what happens is insulin binds to its receptor in the cell membrane. Okay. Next, what happens is activated receptor. After insulin binding, the activated receptor can promote the recruitment of glucose transporters from the intracellular pool. All the transporters are present inside the cell, and these receptor molecules are recruited to the cell membrane. and then glucose transporters. So these receptor molecules are basically proteins. Okay, glucose transporters. These increase insulin mediated uptake of glucose in the cells. These blue ones are the glucose molecules. After the receptor gets lodged onto the cell membrane, then they help in taking up the glucose molecules inside the cell. Then when insulin levels are low in the blood, then glucose transporter can move from the cell membrane back to the intracellular storage pool where they can be recycled, and vesicles fuse to form endosomes. Okay, so what basically happens is these are the ones carrying the transporters. Having these transporters can finally be transported in vesicles forming endosome, and when again these are required, these can be recycled back. Another step, another type of regulation that happens is at the level of pyruvate kinase. Now see here, what is happening is, now pyruvate kinase, what it does is it converts PEP to pyruvate, okay, and releases ATP. Now, pyruvate kinase can have two forms. One is the dephosphorylated pyruvate kinase, which is actually a more active form, and another is the phosphorylated pyruvate kinase, which is a less active form. Now, when the glucose levels in the blood is lower, what happens is the pyruvate kinase is uh, dephosphorylated. Okay, when the glucose levels are low in the blood, what happens is dephosphorylated, like the pyruvate kinase gets dephosphorylated. Now, dephosphorylated one is more active, so it will basically convert PEP to pyruvate. Okay, so so the dephosphorylated pyruvate kinase can activate the conversion of PEP to pyruvate, and we get ATP. Okay, now when it's the other way around, that is blood glucose levels are high, then we have, if the blood glucose levels are high, we have already sufficient amount of energy. So what happens is glucose molecules can then catalyze the phosphorylation of pyruvate kinase. Hmm? Now when pyruvate kinase is phosphorylated, then that is a less active form. So if it's less active, then the formation of pyruvate from PEP would be lesser, and less ATP will be produced in that case. Okay, so this is also one way to regulate the glycolytic pathway. Also, there are many kinds of regulations. Suppose when there's, so these minus ones are the negative regulators of glycolytic pathway, and the plus ones like acetyl CoA, in this case if fructose 1,6 bisphosphate is present, these ones are the positive regulators of these, like particularly these reactions that are taking place in a glycolytic pathway. Okay, now next what happens is, once we have the pyruvate, once pyruvate molecules are formed, then it enters the Krebs. Also, yeah, also glycolysis uh, takes place in the cytoplasm, and then what happens is the pyruvate that was formed, it enters the Krebs cycle or TCA cycle. So this particular cycle takes place in the mitochondrial matrix. So what happens is pyruvate oxidation and the citric acid cycle takes place in the mitochondrial matrix. Okay, pyruvate oxidation, what is happening? Pyruvate is oxidized to acetate and it forms NADH, H plus and releases CO2. Okay, so releases CO2, and acetate is activated by combining with coenzyme A, yielding acetyl coenzyme A. Okay, next, this acetyl coenzyme A, the two carbon, this has two carbon uh, atoms. Okay, the two carbon acetyl group and the four carbon oxaloacetate, which is another substrate in this pathway, can combine to form a 6 carbon substrate, which is a citric acid here. Now this citric acid is converted into isocitrate or isocitric acid, wherein the citrate is rearranged to form its isomer. Basically an isomerization reaction happens. Now this isocitrate gets converted to alpha ketoglutarate. Actually there's another step in between isocitrate to oxalosuccinate, and then yeah, and then alpha ketoglutarate. So what is happening here is isocitrate is basically being oxidized to alpha ketoglutarate, and we get another molecule of CO2 and NADH plus H plus. Okay. Then alpha ketoglutarate again changes to succinyl coenzyme A. Therein, here we see like this 5 carbon moiety is getting changed to a 4 carbon moiety because one CO2 is getting released. Now from alpha ketoglutarate to again succinyl coenzyme A, another carbon gets released in the form of CO2 and we get NADH plus H plus. So alpha ketoglutarate is oxidized to succinyl coenzyme A, forming NADH plus H plus and CO2, and this is almost similar to what takes place in pyruvate oxidation here. Okay, now from succinyl coenzyme A, succinate is formed, which again releases uh, GTP, and that GTP is utilized to form convert ADP to ATP. So again we get one ATP molecule from this pathway. And when succinate is changed to fumarate, then this oxidization results in FADH2 formation. And then when fumarate forms malate, so again it uses up water molecule to form malate, and malate is again oxidized to oxaloacetate, releasing another molecule of NADH, like using another molecule of NAD plus to form NADH plus H plus, and this oxaloacetate can again combine with acetyl CoA to form citric acid. So this way the Krebs cycle pathway goes on. So what is happening here, if we summarize, this TCA cycle or citric acid cycle is taking place in mitochondrial matrix. 2 cycles per glucose molecule or 1 per acetyl coenzyme A, because what we saw is pyruvate changes to acetyl coenzyme A. So one molecule which is a 3 carbon pyruvate molecule is changing to acetyl coenzyme A. So there will be another 3 carbon molecule that was formed from 6 carbon glucose. So 2 cycles per glucose molecule and 3 NADH produced per acetyl coenzyme A, 1 FADH2 produced per acetyl coenzyme A. That means for one glucose molecule, we have 6 NADH and 2 FADH2. 2 CO2 produced via decarboxylation, when we were seeing that from 6 carbon moiety, 5 carbon moiety, alpha ketoglutarate, then succinic acid, 4 carbon moiety. Like that, the cycle was progressing. Then 2 acetyl coenzyme transforms into CO2. So overall, what we are getting here is citric acid cycle inputs are 2 acetyl coenzyme, 6 NAD plus, 2 FAD plus, 2 ADP plus 2 inorganic phosphate molecules, and output 4 carbon dioxide molecules, 6 NADH, 2 FADH2 and 2 molecules of ATP. Okay. Now there's anaerobic respiration also, wherein the pyruvate that was formed out of the glycolytic pathway can directly form lactic acid, 2 molecules of lactic acid. So this is basically a comparison, a diagram showing the comparison between aerobic respiration and anaerobic respiration. So this is the aerobic respiration part, where in glycolysis you have glucose gives 2 pyruvate molecules, pyruvate oxidation forms acetyl coenzyme A, Krebs cycle or the citric acid cycle goes on, and oxidative phosphorylation goes on, wherein which that is that the chain of whatever happens in the inner mitochondrial membrane, the chain of all those protein moieties kept that actually processes the NADH and FADH, 2 molecules that we got from the Krebs cycle, releasing 32 molecules of ATP, and it utilizes oxygen. Now in anaerobic respiration, what is happening? Glycolytic pathway was there. We got ATP, pyruvate. From these pyruvate molecules, either ethyl alcohol and carbon dioxide is released or lactic acid is released in case of anaerobic respiration. Okay. When there's limited or limited supply of oxygen. Now, so this was basically what all goes on, the basic metabolic pathways. Now, how to really grow uh, microbial organisms? Okay, so for that, uh, we need to provide them with sufficient nutrient and sufficient environmental conditions so that they get the appropriate nutrients for their own growth. So that is what we call as a growth medium or a culture medium, wherein we culture the microbial cells. So a culture medium is the mixture of organic and inorganic materials in which microbes are grown. And there can be like different types of culture media which can be divided on different depending on its composition, depending on the state of the medium, growth medium, which can be, liquid, solid, or semi-solid. Okay, and also there can be difference as in distinction or classification based on their functions. So, based on the physical state, the growth medium can be a liquid one. Uh, it should be broth, not broth. It should be broth. Okay. So this is a broth without agar. For example, nutrient broth. Uh, if you guys might be familiar with the term like LB media or luria broth. Okay. Then there's semi-solid medium wherein certain percentage of agar is used, which is a solidifying media, and it has a gel-like consistency. And sometimes when you want to check the motility of microbes, so through, sometimes what happens in case of um, worm like Caenorhabditis elegans, what happens is sometimes there are certain laboratories which check the feeding habits of C. elegans. So they have these agar plates wherein they check the motility of those certain kinds of microbes. So for the microbes to really move, or to check their feeding capacity. Okay, so what happens is if they use a semi-solid medium, the whatever microbes we are trying to study, they can move through the medium and feed, or whatever reward is being kept, they can feed on that particular type of nutrient being provided to them. And there's completely solid medium, which is it can either be 3% agar, 2% agar, whatever. So it is used to check the morphology of bacterial colony. There's what happens is when we plate uh, plating, as in when we culture a certain amount of bacterial cells, what happens is uh, we take a certain amount, maybe as little as 10 microliters of culture, suppose, or 50 microliters of that suspended culture medium wherein the microbes or bacterial cells are growing. And then what we do is on this solid medium, which is primarily made up of agar, on that medium, what we do is we spread it out and smear, like properly spread it out using a spreader or tip or whatever is available. So we spread the culture out, and then we let the microbes grow for overnight, 12 to 16 hours, 10 to 12 hours, and then on that you can see like bacterial colonies growing on the agar plates. Okay, so based on the morphology of the colonies that have grown on the plate, you can distinguish, or it doesn't have to be bacterial colonies, it can be fungal colonies as well. Okay, in fact, on an agar medium, plants are also grown like Arabidopsis, etc. And on the basis of composition, media can be defined or undefined, like chemically defined media in which the exact chemical composition is known, and it consists of chemically pure organic and inorganic compounds which are specific to the nutritional requirements of the microbes being cultured. Then there's complex media, wherein a lot of like we don't know exactly this defined concentration or amount of each constituent that has been added, but overall it can have a lot of a mixture of components like organic components and organic components. And usually what happens is this complex media in general for growing, like suppose culturing large E. coli cells, as in large cultures of E. coli or any such bacteria used for protein prep or some kinds of thing which doesn't have any specific requirement later on, complex media is used, which is actually better for growing the or culturing bacterial cells on a general basis. If there's any specific requirement, definitely then chemically defined media is used. And on the basis of function, general purpose media, the media that supports the growth of a large number of microbes, and enrichment media, the media which contains the substances that prefers the growth of a specific organism that requires more nutrients. Okay. And now, what all what are the common composition or constituents for of a medium, growth medium for growing bacterial cells. Okay, there is uh, it requires amino acids, nitrogen. So the source can be, peptone, which are basically multiple digested peptides. Okay, protein hydrolysate, infusions and extracts. Extracts are, wholesale extracts, like it can be from crushed plant tissues, or yeast extract, like that. That can be a very good source of amino acid or nitrogen. Then growth factors are required, which are obtained from blood, serum, again yeast extract or vitamins. Then there are energy. The bacterial cells definitely require energy to multiply and uh, perform their day to day activities. So energy sources are usually sugar, like glucose, etc., alcohols or carbohydrates. And then there are buffer salts, definitely, because there's a certain range of pH that needs to be maintained for all the activities to be performed. Because what happens, most of the activities are uh, performed by enzymes, which are proteins, which actually need a very specific range of pH, so that they can perform their functions, and basically they can maintain their structure and perform their functions. If the structure gets disrupted, which is very sensitive to pH values, like basically the presence or absence of, as in increase or decrease of H plus ions, then what happens is, if the structure there's some issue with the structure of the protein, then it won't be really able to perform, mostly won't really be able to perform its function. So the buffer salts are provided by phosphates, acetates, citrates, and then there are mineral salts and metals: phosphate, sulfate, magnesium, calcium, iron. And there are certain selective agents like antimicrobials or dyes. You have to sometimes, like, in order to, a particular medium can be, preferred by multiple types of cells. So in order to, or suppose when we are trying to incorporate some kind of, uh, we want to express a particular kind of protein in a cell, how do we know that whatever we have uh, put inside the cell of our interest, that has actually gone, or basically the plasmid has actually gone in and fused with the genome of the, as in that has gone inside the bacterial cell, and it will be expressing our protein of interest. So for that, certain dyes are used that can demarcate between what all cells have actually taken up our plasmid of interest and can express our proteins. Okay, there are indicator dyes like phenol red, neutral red. So these are, phenol red can be used as a pH indicator. Then there are gelling agents, one we already saw, agar. Then gelatin can be used, silica gel can be used, or alginate can be used. And this is again a very broad table of what all compositions are there, what are the commonly used growth media for growing bacteria. So see, there's LB media, which is a very frequent and very commonly used in laboratories for protein prep and all those, like cloning activities and all. And there's like yeast, peptone, tryptone. This media is there. SOC medium, SOB medium. These are also there. So there are different kinds of growth medium, which are used ba based on the type of application that is required, or the downstream processes that we want to uh, use the bacterial cells for. Next, there's yeast medium. So these three slides, as in these two pictures, have been taken from the lecture slides only. And this is again yeast medium. What all media are used for growing yeast cells. And then this mammalian cell culture medium. So in mammalian cell culture, what happens is, now during mammalian cell culture, we have to be the person handling the mammalian cells during cell culture have to be those people have to be really cautious and really hygienic, like maintain really good lab practice, because these are very much prone to getting contaminated by bacteria or fungi, because these are these can, like, bacterial cells require like very little as compared to mammalian cells, in a cell culture, uh, bacterial cells require like minimal nutrients for their growth as compared to mainly, and also maintenance, minimal maintenance as compared to mammalian cells in a cell culture uh, medium. Okay, so what all have to be taken care of? pH of the media, again, that should basically, this is the range should be lying within the physiological pH range, and filtration should be performed at low speed. Okay, so what happens here is the media that has been that liquid, this particular liquid, has to be filtered to remove any particulate matter, and the filter paper through which these types of media are filtered, those have pores as small as zero point 0.2 micrometers. Okay, so so that like most of the contaminants can be left out, and we get as like a pure, as much as possible we can get a pure or decontaminated growth medium. And this filtration should be performed at low speed, because if we are performing the filtration, if we are filtering the media in a very at a very high speed, then what happens is, when uh, it can enlarge the pores. It the size is around 0.22 micrometers, and that pores of the filter paper can get enlarged when we are trying to filter the whole sample or whole medium via that small pore size. Then the pore size can get enlarged, and what will happen, that the contaminants that were supposed to be left out can get inside through the filter paper. And then serum is used, which is heat inactivated, to remove certain other, like there are antibodies, there can be antibodies, etc., which might be problematic in the downstream processes. That has to be inactivated. Otherwise, the other organic substances, other organic requirements for the cell to grow, can be found in the serum. Then antibiotics, to remove unwanted, to select for whatever required, uh, whatever cells are required, to be grown, and to suppress or just to kill the growth of any other unwanted cells in the medium. And contamination checking, same thing. Contamination checking can be done, this usually the cell culture medium has phenol red dye as an indicator of suppose pH change. Now, if pH is changing, suppose if there's bacterial growth in the cell culture medium, then what will happen is, usually like when bacterial cells grow, the they release acidic byproducts. Okay, so bacterial growth can change the phenol red color to yellow. Okay, and when if the medium becomes alkaline, which usually happens, again, there are some other cases also, but generally what happens in case of suppose fungal growth, the medium becomes alkaline, and phenol red can become uh, pinkish kind of. Okay, so these indicators can be used to get an idea of any contamination that might be present, or contamination can also be checked under microscope, wherein you can actually see the morphology of any other, whatever mammalian cells you are looking at. If there are other cells having very different kind of morphology, or some bacterial cells are motile. Okay, so those can be visualized under a microscope, have moving. Okay, so again, so these are some of the ways to check for any type of contamination present in a cell culture medium. And these are some of the common types of cell culture media, like EMEM. This is Eagle's minimal essential medium. This was the first minimal medium to be developed. And there's DMEM, similar to EMEM, but having higher concentrations of vitamins and amino acids. Then there's RPMI 1640. It is a more complex one, which is used to grow human lymphocytes and can also support a wide range of cell types. Then you have this IMDM. This is again a highly modified version of DMEM. So these are the overall commonly used uh, cell culture medium, out of which DMEM is really common in laboratories. But again, the type of medium that you need to use depends on the type of cells you want to culture, what all things you want to investigate using those cells. All of it depends on uh, the choice of the medium depends on your objectives that you are trying to look at, or what all experiments you are trying to perform. Okay. Now, what all precautions need to be taken while preparing bacterial growth media? So basically, what mainly the precautions have to be taken so as to make the growth media as sterile as possible. Okay, so that now suppose E. coli cells are usually cultured in LB media. Okay, now that what people usually do is, so when LB media is prepared in flasks or glasswares, they have to we like usually what people do is they plug it using a cotton plug and then autoclave it at very high temperatures at around 121 degrees Celsius and high pressure, and then so that intense heat and pressure can actually eliminate most of the other like unwanted microbial growth, so that in that particular growth media when we add our inoculum of interest, that will only have those cells that that are of our interest, and any unwanted content is removed. Okay, then also when you prepare, so this is applicable for any type of media that you want to uh, prepare, like whether it's agar medium, which is a solid growth media, liquid media, all of these have to be autoclaved and in like properly and have to be maintained under sterile conditions. Then um, suppose when you are dealing with microbial microbes, then it's always a good thing to wear gloves and wipe things with ethanol, crude ethanol, which is again which adds to the and sterility of your workspace. And then also, so when you are preparing agar medium, solid medium, so there are those petri plates that are used, and then uh, we study the growth of, or we let the bacterial colonies to grow on the agar plate. So what happens is sometimes, uh, it's not sometimes, most of the times what happens is we add an antibiotic so that only the bacteria of our interest grows. Okay, so now suppose um, maybe some kind of gene that we introduced has an additional ampicillin resistant gene attached to it, so that we can select for those cells which are ampicillin resistant. So if those cells which are ampicillin resistant will definitely have our gene of interest attached to it. Okay, we expect that. So only those, so if we put in an agar medium, if we put antibiotic into it, some ampicillin, what we were talking about, then what will happen is only the cells that has taken up that particular gene which codes for antibiotic resistance, and that antibiotic resistant gene is attached with the main gene of our interest. So that means if a particular cell is growing on that medium containing ampicillin, so only those cells are able to grow that have become ampicillin resistant. Okay, so this way we can select for the particular type of bacterial cells. Now, while preparing such kind of agar, what happens is, because when we are autoclaving the stuffs, it's like at 121 degrees Celsius. It's really hot to handle, and it's always good to let the pressure come down. Let the You shouldn't be opening autoclave. Basically, is like a pressure cooker. Okay, so you shouldn't be really opening the pressure cooker, and also it is actually dangerous to open a pressure cooker in general when it's at high pressure, right? We should let the steam come down. We should let the thing cool down a little bit, then only we can open it, and we should be opening it that way only. And then when it's hot, you shouldn't be really adding the antibiotics. Suppose we were talking about ampicillin, because that will degrade the whole molecule, the antibiotic molecule. So it's a good practice to let the temperature of the particular medium, particular solution come down a little bit, and then when it's at at room temperature, then not only room temperature, maybe a little higher than room temperature is also okay. Then add the antibiotic and then mix it properly. It should be mixed properly, and then we should let it cool down. Then we shouldn't wait for too long for the whole preparation to cool down, then it will get solidified. Then there's no point in adding the antibiotic after that time, because it will be like there, it doesn't make sense at all. Okay, so there should be a proper uh, time point wherein you should be adding the antibiotics when the solid medium or agar doesn't get solidified, or it it isn't hot enough to inactivate or degrade the antibiotic molecule altogether. So these are some of the precautions that need to be taken when we have to prepare any type of growth medium for culturing bacterial cells. Now, what all types of um, pro- mainly we will look at the asexual type of reproduction in bacterial cells. One of them is binary division, and then what basically happens here is the whole parent cell, one of the parent cell, uh, basically divides into two daughter cells. Okay, so what happens is cell replicates its DNA. There's this cell wall, cytoplasmic membrane or plasma membrane, and the DNA has replicated. Now, the cell becomes elongated. Okay, the cytoplasmic membrane elongates, and the DNA molecules move to kind of opposite poles. And then there's an invagination of the cell wall, and this cross wall can form the cell membrane can invaginate, and finally the whole cross wall is formed completely, having two sets of DNA. Okay, and then these two cells divide and form daughter cells. This is the simple binary division in bacterial cells like E. coli, etc. Then there's budding also, wherein what happens is a bud begins to form on this is the parent cell. A bud starts forming, then the nucleus or the after the genome gets copied or duplicated, that particular one moves into the bud. Okay, nucleus copies and divides, the bud receives one copy after the nuclear division or the genome, and after the DNA replication, and then bud now can become a separate daughter cell. Sometimes what happens is chains of cells can uh, be formed, like this chain is formed, chain of cells is formed on this parent cell. And also many times what happens is this bud, that the newly formed bud, pinches off to form an independent entity on its own. Okay, then there's fragmentation also. So this is mostly seen when the conditions are unfavorable for the bacterial cell to grow in. So what happens is mostly during unfavorable conditions, the bacterial protoplasm, what we read, protoplasm means cytoplasm plus nuclear, whatever is inside the cell membrane. Bacterial protoplasm undergoes compartmentalization. See, and subsequent fragments are formed, which are on their own minute bodies, which are called gonidia. And when the conditions, so like favorable conditions for the bacterium return, then each gonidium grows into a new bacterium. So it becomes apparent that prior to fragmentation, the bacterial genome has to undergo repeated replication so that each fragment gets a copy of it. So when the unfavorable conditions set in, what happens is it has to really work a lot or use up a lot of its resources so that it can tide over the unfavorable conditions. And when the conditions return, as in favorable conditions are there, then each of the fragment that has a copy of the genome, that can form a whole new bacterium. Okay, now if we look at the bacterial growth curve, basically what happens is it goes via three main stages. After that, death phase occurs wherein bacterial cells start to die. So the first phase is the lag phase. What happens here is this is the very beginning phase in a bacterial over in the whole bacterial growth phase. This is the initial phase wherein a lot of cellular activity is going on, but overall growth is not there. Growth as in division isn't taking place. In suppose if we take the example of E. coli, then E. coli isn't basically dividing, but then a lot of uh, and the cells are metabolically active. So what a lot of enzymes which are actually crucial, like really crucial for later stages of its like division, etc. So such enzymes are mainly synthesized, and then cells increase in size because a lot of metabolic activity and synthesis of these crucial enzymes, etc., are taking place, but no cell division occurs during this phase, this particular lag phase we are talking about. And cells are actually busy synthesizing a large amount of protoplasm before moving on to the next phase of active multiplication. Next, when they're actually dividing, which is this phase of the exponential phase or the log phase. Okay, now what happens in exponential or log phase? Cells are actively dividing and doubling in numbers after each generation time. So doubling in numbers, as in if we talk about basically the binary fission or whatever we saw, binary division in bacterial cells. So from one parent cell, what we are getting is two daughter cells. Now, for the first generation, two daughter cells will each give rise to two more daughter cells. So overall, in the second generation, we'll be getting four daughter cells. Okay. So similarly, all these types of divisions occur in exponential phase. Why exponential? So after each generation, suppose if the generation number is x, then the total number of cells that we are getting finally, the daughter cells from one parent cell, it comes up to 2 to the power x number of cells. Okay. So like suppose from 1, 2 are coming, from 2, 4 are coming, from 4, like from 4, each of the 4 daughter cell will be will be uh, giving rise to two more daughter cells. Okay, so in this way it goes on. How much ever generations we are trying to look at that, so each particular daughter cell will be giving rise to two more daughter cells. So this is an exponential division wherein number of daughter cells are um, represented by 2 to the power x or 2 to the power n, wherein x or n represents the number of generation. And yeah, definitely, if the cell is dividing, the metabolic activity is really high at this time. DNA, RNA, cell wall components, and other substances which are really necessary for growth are being generated for division. And during this phase only, what happens is when there are antibiotics which target the microbes, or disinfectants, where we use disinfectants to remove any contamination like microbial contamination. So most of the antibiotics and disinfectants are designed in a way that um, actually target the bacterial cells or microbial cells in this particular phase wherein they are actively dividing. So if they are targeted at this particular phase, uh, then their growth can be minimized, and ultimately can be totally finished off. So what happens is during this time, bacterial cell walls can be targeted, which like specific antibiotics are there. Um, mm, Ampicillin is one of the antibiotics which targets the bacterial cell walls. Then there are different types of antibiotics which target the transcription machinery. Definitely, transcription is formation of RNA from DNA. Okay, so if RNA needs to form, so that RNA can further code for proteins. And these proteins are utilized by the cells to carry out their metabolic reactions, to carry out the division activity or multiplication activity. Now, if those particular machineries are targeted, then those processes or those pathways are targeted, definitely their numbers can be reduced to a large extent. Okay, so like there are antibiotics like um, tetracycline, kanamycin. These target the translational apparatus, basically the ribosomes that are involved in, reading the messenger RNA and coding for amino acids. So there are different kinds of range of antibiotics that specifically target certain processes so as to suppress the growth, suppress this exponential growth of bacterial cells. Because in E. coli, their doubling time or each generation, so one cell gives rise to two daughter cells within 20 minutes. So just imagine like within 2 3 hours, or maybe whatever I was telling, like when we want to see the bacterial colonies, when when we want to grow bacterial colonies on an agar plate, what happens is we usually incubate it for 10 to 12 hours or 12 to 16 hours. So just imagine the number of cells that are present in that particular broth that we have cultured for 10 to 12 hours. Okay, because all of these are going undergoing exponential growth after every 20 minutes. Okay, and then there comes stationary phase. Ideally, this should have uh, been like this, wherein this is the theoretical growth curve. But what happens as the cells go on dividing, they are using up the nutrients that are being provided to them. So, after a certain point of time, the number of cells are too high, and the nutrient source or the resources that are available in the medium become limiting. Definitely, after a certain point of time, it will become limiting. In an ideal scenario, this would have been the case, but in a natural scenario, what happens is the available nutrients or resources become limiting. Definitely, as the cells are too high in number, competition for the nutrients or the resources available increases, and also because they are multiplicating at such high numbers, multiplicating at high speed and high numbers, the waste products can start to accumulate. So what happens is now the cell growth reaches this plateau phase or stationary phase, wherein the number of cells that are dividing versus the number of cells that are dying becomes equal. So overall, we don't see a growth in the number of bacterial cells or microbial cells. So cells become less metabolically active because whatever activity was there, it started, but then the competition or the paucity for resources came in, and uh, they couldn't really divide with as much efficiency as that was seen in the exponential phase. So what happens during this stationary phase is usually spore forming bacteria produce endospores in this phase because nutrient or resources become limiting. So what happens is this is actually an unfavorable condition for the bacteria. So those bacterial cells which can form spore, they produce endospores in this phase. And also pathogenic bacteria can actually in this particular phase, because there's competition now among all the cells to take up the resources or use up the resources, what happens in this phase is the pathogenic bacteria can generate certain substances or virulence factors that can actually help them to survive harsh conditions. And these virulence factors are what lead to different kinds of diseases in plants, suppose in animals, in humans, etc. Then finally, there's death phase. Again, this is an exponential phase wherein the number of dividing cells are much less than the number of dying cells. Okay, nutrients are kind of diminished and waste products are accumulated to a large extent. So the number of dying cells rises and number of living cells decreases exponentially. So there's a sharp decline in the growth. Okay, so that's why. So ideally, uh, can someone tell me that suppose when we need a large amount of proteins from the cells, so what should be the ideal condition for growing the bacteria? Well, at which particular phase can we really harvest the bacterial cells? Anyone can try. Yes, yes, that is when they are actively dividing. Thank you, Mona Lisa, right? Yeah. Yeah. So that is when we usually harvest the cells so that we can get maximum amount of proteins at that phase. Also, so this particular exponential phase that we had discussed, when we culture, how do we know what are so there should be a particular value assigned to, uh, when the so in a culture broth, what happens when bacterial cells are dividing? So first, the broth is totally clear. It's a transparent solution, like yellowish transparent solution in case of LB media. Now, when cells are slowly growing, what happens is the solution starts to become turbid. Okay, the transparency decreases. So what happens is each time, after each time point, maybe after an hour or after 30 minutes, what happens is the measurement absorbance values are taken, okay, at certain wavelengths, wherein if the value, if the media becomes more turbid, then the value of that absorbance will increase definitely, because the transmittance will be very lower of that particular light through the medium, because now there are cells that will hinder or interfere with the passage of the light that is being provided through the solution. Okay, so based on those values, so in the exponential phase, when ideally when should we be harvesting the cells is when that absorbance value is in the range of zero point 0.5 to zero point 0.8. So what happens is this is this part of the phase, okay, this part of the phase when cells are exactly in the middle of the exponential phase. So sometimes what happens is if we harvest the cells at this point, it comes to around maybe 0.91 something around, and around that part, what happens is wastes, again what we saw, like waste materials start to accumulate, and then uh, basically there might be some other uh, protein stuffs, or there might be more dead cells. So we don't want that. We want actively dividing cells, and we want to harvest those particular cells which have enough amounts of proteins. And we had introduced our gene of interest into those cells to harvest, like get a lot of proteins of our interest from those cells. So we actually want to harvest the cells at that particular area. So in the mid log phase or mid exponential phase, this is the ideal condition when the cells should be harvested, when the bacterial cells should be harvested. Okay. Now, how can we really measure microbial growth? So basically, one is microscopic count. What we basically do is, suppose you take one type of dish or plate, usually petri dish, which are transparent ones. You put some amount of culture. Suppose if on a growth medium, which is a liquid growth medium, you can take few microliters of that growth medium on that plate, or on a slide also, microscopic slide also you can take, and then you can count the number of cells just visually under a microscope, and like manually only you have to count the number of cells. Now here in this case, what we don't want is the cell number shouldn't be too less. Then it is really an inaccurate measure of the number of cells in the culture. And again, it shouldn't be too high. Like if the solution has become too turbid and we are like directly taking that particular solution, like few microliters from that particular culture which is too turbid, and then trying to look on the microscope, then it's too crowded. Okay, the field of view will be too crowded, then there might be overlapping cells, and the count will be again inaccurate. So what usually people do for microscopic counts is they visual like basically we dilute it using the same medium, and then try to count the number of cells. And when we are really calculating the stuff, then like we take into consideration the dilution factor, how much by how much times we had diluted. Then if we multiply using that dilution factor, that will give us the number of bacterial cells in the whole culture. Okay, whatever volume of the culture is, suppose it can be 5 ml, 1 ml, 10 ml, whatever. Or it can, for protein preps, actually it can go up to 2 liters, 3 liters culture, wherein you need a lot of proteins for further downstream assays, whatever experiments you want to perform. So, uh, yeah, for microscopic count, this is what is happening. Like, this is what I was talking about, the pour plate. So, use pour plate or spread plate for viewing under a microscope. Concentration shouldn't be too much because overlapping cells will give inaccurate counting, or it shouldn't be too few, which is again an insufficient number for accurately determining the number of cells. So this is basically showing that test tubes which are subsequently diluted. Suppose this is actually called serial dilution, wherein from one test tube, suppose you have 1 ml of the original turbid culture, say, having microbial cells, and then from here you take few microliters into all, suppose 5 ml media, and then from here you again take 1 ml in another 5 ml media. So this way you are actually diluting the cells and ultimately seeing them or visualizing them under a microscope. Yeah, so this is what happens after dilution, physically counting the bacterial cells under a microscope. Okay, and definitely this is there, like you should be keeping a count or keeping a log of whatever you are seeing so that you don't lose, because this is a manual thing, you don't have any record other than your memory. So it's good to write it down. Then there's again serial dilution and plating. So in serial dilution, again you have a series of test tubes. So this is the original inoculum. Suppose you took 1 ml and in a 9 ml liquid medium, the same medium wherein the bacterial cells were grown. And then if 1 ml original inoculum goes into 9 ml broth, then it is 1 is to 10 dilution. Now from the, and also definitely, now imagine in a 9 ml broth, you are putting 1 ml of solution. And if you immediately take 1 ml out of it, then it would not make as much sense, but uh, because it isn't properly resuspended into the whole broth. So what you have to do after each addition, you should be really, like there are pipettes to do that. If it's a closed tube or something, then you can definitely slowly mix it properly. So this mixing should be done. After that, you take suppose 1 ml again into 9 ml broth. So what happens is there's 1 is to 100 dilutions. So in the same way, you take multiple tubes having clear LB broth and add 1% as in 1 ml of inoculum into each test tube, resulting in see, now it's around 1 lakh times diluted. And now from each plate, now each culture, what you do is, this is the plate I was talking about, what we used to see the morphology of bacterial colonies, uh, or count the number of bacterial colonies, or there are uh, many other applications of plating bacterial cells and getting the number of colonies from there. So what happens is, if this is only 1 is to 10 times diluted, see, this will give us a lot of a lawn kind of thing, uh, a total smear wherein we don't have single colonies to count. So you can't really count the number of colonies from here. And subsequently, when it's getting diluted to such large numbers, now see, it's very prominently 4 colonies are there. Okay, so this is an example, like number of colonies on the plate into reciprocal of dilution of sample gives us the number of bacteria per ml in 1 ml of culture. So now suppose 32 colonies are found here, suppose in this particular plate, like 10,000, 1 is to 10,000 dilution. Then 32 times 10,000 will give us this many number of bacteria in only 1 ml of sample. Okay, so if you have cultured like around 100 ml uh, of if you have cultured microbial cells in 100 ml of sample, then just imagine the number of bacterial cells that you can actually have and harvest for downstream processes. Okay, so serial dilution is this one, and plating is basically you take this thing and spread it out on this particular growth medium, which is a solid growth medium ideally. Okay, mainly agar, that is used on a very regular basis in laboratories. Next, you can use use turbimetric method to measure the turbidity. What is happening here? The same thing like you had in the serial dilution method. You are diluting basically to reduce the turbidity. Okay, the more number of cells are there, the more turbid the solution will be. So what happens is, if a clear solution is there, suppose uh, the LB media that we are talking about, so light is passing through it because there's nothing, no kind of entity to really obstruct the path of light. There will be a certain reading. Now, with reference, now blank is a reference. Okay, with respect to that, now the readings will be taken for the subsequent samples. So when you really blank it, the reading on the spectrophotometer comes to 0. Okay, now when you shine light at a particular wavelength, in this case, in case of seeing the bacterial density, bacterial growth, the 600 nanometer wavelength of light is used and absorbance values are recorded. Now, when there's bacterial cells in this particular medium, what happens is definitely see uh, much less light is passing through and getting detected by the detector as compared to the ones wherein most of the light can pass through. Okay, so the transmittance gets really decreased in a bacterial suspension. So the more number of cells present in the suspension, the lesser will be the transmittance, lesser will be the passage of light through this particular solution, and higher will be the reading of the spectrophotometer. Okay, because transmittance, if you remember from, there's a Beer-Lambert law which uh, which draws a relation between the absorbance of the particular sample we are measuring and the concentration of the sample. So it is directly proportional. A, or absorbance, is directly proportional to the concentration of the sample being uh, tested. Okay, so what happens is it is directly proportional. So as the concentration of sample increases, here in this case, the number of bacterial cells in the medium, then the absorbance value will increase. Okay, and in case of bacterial medium, the wavelength that we use to detect the absorbance is 600 nanometers. Okay. Similarly, suppose this is for bacterial cells. Now for protein concentrations. Okay, for measuring protein concentration, another type of wavelength is used, which is 280 nanometers, because that is a particular wavelength wherein there are aromatic amino acids called tryptophan, phenylalanine, uh, uh, tyrosine. These are the ones, due to their uh, electron cloud on the benzene ring present, they can give absorbance, they can absorb light at 280 nanometers and give certain readings. Similarly, for nucleic acids, DNA or RNA, there are purines and pyrimidines, right? So those are again like nitrogen and oxygen containing rings having doubly bonded regions having electron clouds spread over those rings. So that again absorbs light at 260 nanometers, and we can again that is the same thing. It is also concentration dependent. Okay. So now for measuring microbial growth, what we saw, microscopic count, we can count the number of cells directly like physically counting the number of cells under a microscope. We can measure the turbidity using a spectrophotometer by measuring the absorbance of the sample. Then there's wet and dry mass, wherein basically when I was talking about harvesting the cells, okay, in the exponential phase. So what we basically do is, there are the cultures, okay, culture of bacterial cells in a test tube. Now, though the whole solution, the whole suspension is centrifuged at high speeds. I think in the first lecture we saw how to really, harvest bacterial cells. We suspend it at high speeds, then we get a supernatant and a pellet. So pellet is basically all the bacterial cells, and we have actually harvested the cells. So that is what gives us the wet weight. Okay, now if the cells, those particular cells, the bacterial pellet that we got, those have numerous, like millions of cells in the pellet, and if that pellet is actually dried um, properly using certain uh, apparatus, and for a certain period of time, then we get the dry mass. So that can also give us an idea with respect to, there should be a reference point which should be used for comparing the weight obtained after the bacterial growth, or suppose wet and dry mass. If that pellet, we take the, suppose we got the pellet in a particular tube, the empty tube's weight plus the tube plus the pellets weight, as well as after drying the particular pellet, then the weight that has been obtained can give us the dry mass. So in this way, there are various kinds of methods to really measure the microbial growth, most commonly used one is, the absorbance one, spectrophotometer one, or microscopic count is also commonly used one, wherein there are not only that one, like we were talking about only putting few microliters of the cell culture on a plate and directly looking at it. There are certain kinds of apparatus, like I don't know if you're aware of it. You might be aware of it, which is hemocytometer. There are certain grades of defined sizes, defined volume, okay, defined height and area, wherein you can calculate the you again you have to dilute your samples at a particular range wherein you are able to count the cells. It shouldn't be too high or too low. And then you actually, again, based on the volume, height, area used on the grids, you can calculate the number of cells present in the whole culture. Okay, so there are different ways to do to measure bacterial growth or the number of cells present in a bacterial culture. Okay, so now we move on. Look at some of the questions that uh, that are very much related to the assignments that you are given during after every week's lecture. So the first question is, which of the following is the cofactor of hexokinase? Hexokinase was the enzyme that was used in the glycolytic pathway. Okay, the very first enzyme. Hmm? So can anyone take a guess, or if anyone knows? Okay. Yes, Mg2 plus is a cofactor of hexokinase because it needs the presence of this magnesium ion to really perform its function, to perform the perform its function in the glycolytic pathway. So if the term cofactor has come in, let's just very briefly look at what cofactors are. So there are many enzymes which require these cofactors to actively perform their function. Okay, uh, now this enzymes are basically proteins which are ideal like predominantly globular proteins. So this is lysozyme enzyme wherein it doesn't really require anything to get attached to or bind to. It can really perform its function on its own. But now cofactors are a non-protein component of an enzyme, okay, which can be organic molecules. When such non-protein components are organic molecules or coenzyme, those are called coenzymes, or certain inorganic ions that we are seeing in this case, like Mg2 plus getting added to hexokinase, Mg2 plus getting added to hexokinase, then this is an inorganic ion that is required by hexokinase to perform its duty, as in perform its function. So enzyme plus prosthetic groups are permanently attached ones. Okay, cofactors again can be permanently attached in case of prosthetic groups in which, yeah, these are permanently attached. So in the case of flavoprotein plus FAD, if unless this is attached, it won't be performing its function, and there are temporarily attached coenzymes which detach after a reaction and may participate with another enzyme in some other metabolic reaction or metabolic pathway. Okay. So the next question is, in glycolysis, two ATP molecules are utilized by the enzymes dash and dash. So basically we are talking about the preparatory phase. Okay. Like ATP molecules are getting utilized and ADP is given out. So, does anyone remember the name? Yes. Yes, very nice. So, this is the same pathway. Uh, See, hexokinase is requiring one ATP to phosphorylate the glucose molecule and another ATP required by phosphofructokinase to really phosphorylate the fructose 6-phosphate molecule. Okay. Now, fructose 1,6-bisphosphate is broken down into glyceraldehyde 3-phosphate and DHAP by which enzyme? Oh, very good. Now, which of the following cell surface receptor is involved in the uptake of glucose? Can anyone take a guess or anyone knows? See, insulin receptor will take up insulin. All right. So here we are talking about with yes, yes. Okay. Now, during the event of low blood glucose, like we just brushed it up in the previous slide, the enzyme pyruvate kinase is when the blood glucose level is lower, what happens? Can anyone recall? Yes, yes. Can you repeat? I think you were. Yes, yes, yes. True, because when it's dephosphorylated, that is the active form of pyruvate kinase, right? So when it's active form of pyruvate kinase, then PEP won't be, as it will be readily converted to pyruvate to give ATP. And when it's phosphorylated, then it should be, uh, when it's phosphorylated, it is less active. है ना? So this is the same pathway that we are looking at. Okay, the same slide, the same thing whatever we read earlier. Okay, now this is, I think this will be, everyone will be able to answer. One molecule of GTP is equivalent to dash molecule of ATP. Can yes, yes. Okay, answer is A1. Which of the following pairs positively modulate the conversion of isocitrate to alpha ketoglutarate in the, what we are talking about is the citric acid cycle. Okay, so can anyone take any guess? It's okay. If you know, it's well and good. If you don't know, that is also fine. Any guess or any idea? So ADP, yes, yes. Okay, so what happens is, this is basically referring to the enzyme that is involved in this particular conversion pathway of isocitrate to alpha ketoglutarate. Okay, so this is what is happening when isocitrate is getting converted to alpha ketoglutarate. In between, there's isocitrate dehydrogenase or IDH enzyme, which is actually activated by the presence of ADP, citrate, and calcium ions, and it is actually inhibited by NADH, NADPH, and ATP molecules. Okay. Next is production of alcohol under anaerobic conditions in yeast is facilitated by the enzyme alcohol dehydrogenase, which actually converts acetaldehyde to ethanol. Substrate pyruvate binds to the active site of the enzyme with the help of a dash ion. Again, this is, yes, very good. This is again a cofactor that we are talking about. Okay. Which of the following is the function of phenol red in DMEM? DMEM is the mammalian cell culture medium that we talked about in the previous slide. So what was actually the function of phenol red? Anyone can take a, yes, yes. So can you tell like what might be the color changes? Like suppose if it's acidic, what might be the change in color of phenol red? Yes, yes. Okay. So yeah, detect change in pH. Now, in a Petroff-Hauser counting chamber, the volume of each chamber is approximately, so actually this is Petroff-Hauser counting chamber is again depending on my, like we looked at the different types of methods used to count the number of cells or microbial cells. This is another one, another type of apparatus or a particular type of chamber, like I was telling about hemocytometer. So another one is Petroff-Hauser counting chamber. Now this is overall a very technical question wherein it depends on the dimensions of the chamber. Can anyone take a guess? Or if one has gone through the lecture videos? Haha, tell. Yes. So it will be 1 by 20,000 millimeter cube. Okay. So just go, we can go through the Petroff-Hauser counter. What happened? This is again from the lecture slide only. What happens is this has the, there are these squares, okay, which have a particular area and a particular depth, which is around 1 by 50 or 0.02 millimeter in height and has 1 by 400 square millimeter of area. Okay, so what will happen is the volume will give us area into height. We'll get this volume 1 by 20,000 millimeter cube. And so what happens here is, this is an example to solve the question, because I think in the assignment questions, this demo assignment question, there are two questions related to this particular counting chamber. So if each chamber has 8 bacteria, okay, so if each chamber has 8 bacteria, then there will be 8 into 20,000 millimeter cube of bacterial cells per millimeter cube. Now, if like for 1 millimeter cube to ml conversion, 1000 times, it's 1000 times, right? So that's why 20,000 into 10 to the power 3, that gives us 1.6 into 10 to the power 8 bacterial cells in 1 ml of culture or 1 ml of suspension. Okay, it's the same thing that is written here. A very high or low concentration of bacterial sample cannot be counted accurately. Okay, so this is the, yeah, this is what was the question. What was the volume of each chamber in Petrov-Hausser counting chamber? This is option C, 1 by 20,000 millimeter cube. Now it's basically the same question that we saw in the previous example. If each chamber of a Petrov-Hausser counter has 5 bacterial cells, then the total number of cells per ml of the culture will be how much? Can anyone take a guess? Or just tell the answer maybe. It's like simple calculation, what we saw in the earlier example. In that, it will be only 10 to the power 8, right? Because what was the volume of the chamber? 1 by 20,000, right? So if there are 5 bacterial cells, no? So then 5 into 20,000 into 1000, है ना? So it will be these many. Yeah, but the approach was perfectly correct. Okay. Okay. And this is, I think, uh, everyone can answer correctly. What will be the correct sequence of the different phases of bacterial growth? Last will definitely be the death phase. Option B. Yeah. Lag, exponential, stationary, and death. Okay, option B. Now, in which of the following phase the newly divided bacterial cells derive nutrition primarily from the older dead bacteria in the culture? With yes, 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 yes. Take a guess. It will be stationary phase, because what happens is in the lag phase, uh, ideally those are like very the first initial phase when the cells are metabolically active and it hasn't divided yet. But what happens is when slowly when the cells are dividing, that is the exponential phase. Now in the stationary phase, what was happening is number of dead cells and number of dividing cells were equal, right? That's why we don't see a population growth in the microbial culture. So what happens is, if now if the cells are dying in that phase, definitely cells are dying because the nutrition becomes limiting, as in the nutrients or resources become limiting. If in that phase the cells are dying, then definitely if the cells are dying, they will uh, if the cells are dead, there are certain, if the cells are somehow broken or opened up, then there will be already formed nutrients or something present already, but again, uh, which can be taken up by the, which can act, those particular dead cells can act as as a source of nutrient for the live cells. Okay, for the cells which aren't dead. But also, yeah, there's another thing that should be considered when we are discussing this question, is that when cells are dying, those cells can also give rise to certain types of proteins, which are like predominantly those won't hamper, but there are certain accumulation of proteins which can be detrimental for cell growth. But if we talk about in a general sense, newly divided bacterial cells can derive nutrition from older dead bacteria. Wh- uh, which phase, in which phase actually we are getting a lot of older dead bacteria? That is the stationary phase. Okay, so the correct answer would be stationary phase, or option C. Okay, now a bacterial culture medium was inoculated with roughly these many number of cells, bacterial cells at 10 a.m. and allowed to grow for 12 hours. So can you tell the number of generations the cells have grown in the given time span, considering there was no lag phase? Like if we consider only exponential phase has taken place. Okay. Just you can take your time to calculate and let me know the answer. Basically, what we want here is the number of generations. How many generations, for how many generations the cells have grown. If they have been allowed to grow for 12 hours, given that their generation time is 20 minutes. Can someone try to answer? How did you come to that answer? You can just, this is a discussion session. No need to like take pressure and all, so that we can together. Yes, it is coming to 36 because what will happen is, see, the cells are growing for 12 hours. Now see, the 10 a.m. time, this is not required. 5,000 cells, that is just given to maybe just confuse you. That is the primary number of cells, or like the initial number of cells. Okay. Okay. Yes. Okay, nice, good. So 12 hours, basically if we convert hours to minutes, it's 12 into 60. And if one generation time is 20 minutes, then it will be 12 into 60 by 20, which will give us option D, 36. Okay. Now in the turbimetric method, whatever absorbance calculation that we looked at, the OD refers to the optical density, or basically the absorbance of the bacterial culture is measured at what will be the value, 400, 500 to 2600 nanometers. What will be the wavelength? Does anybody remember? 600 nanometers. Yes, that is where that is the wavelength where we actually, this is the, that's the wavelength where we try to measure the optical density. Okay, so that was it for today's class. Stop presenting. Okay, so we'll meet again the next week on Sunday at this time only from 3 p.m. and we'll discuss the next uh, set of lectures, just summarize the lectures and then look at few of the questions. Okay. Okay, see you then. Chalo. Bye bye. Thank you so much for joining the meeting.